So thanks a lot. I'm really excited about this opportunity of presenting to this group of trialists and methodologists. I believe that there is a lot that I would like to learn from all of you, and hopefully my presentation is going to be a bit of a provocation to a dialogue, and I would appreciate as well if there are opportunities or interest from you or other colleagues to reach out to me for further discussions after this meeting. So a few days ago, I presented at the IMDRF forum in Japan, and I started with these driving questions, which I think are the same ones that I'd like to put to you as what is driving what we are doing in this community of in silico methods. One is, how do we provide safety, quality, and efficacy and durability measures of products that we have, particularly when we have a global market for these devices, and most of the times, regular trials might be actually limited in the geographies they really cover, depending on, the trials that have been implemented at a given point in time. Also, how we manage the risk and opportunities when new technologies come, technologies like in silico trials themselves, but also technologies like new medical devices and new medical products that are coming to market. And also, how do we help our regulators that we collaborate with to gain confidence in engaging with them as part of the review process? So this is going to be a bit my vision, my outline of the presentation, and I'll try to cover this in about 40 minutes with the idea that we have at least ample time for discussions afterwards. The first thing is I would like to start by explaining where I think we are today, and therefore where the imperative for actions come from, and what happens if we do nothing as an alternative. So there is, in my view, a fundamental market failure at the moment in the area of in the medical products domain, in the sense that regulators are aware that what we are doing at the moment has limitations in terms of generating evidence, and I'll give some figures in the next couple of slides. But also, and so is the case for the medical, say, medical device or pharmaceutical industries. But it's easier to continue the way we are than actually, moving away from it, because there are risks in both exploring something new and risking continuing in the way we have at the moment. So there is a need to actually have ways to put together these two communities and the academic communities together to actually figure out a new way forward. But if we don't do that, we are having issues both from ethical aspects, considering the use of humans in trials, but also animals as an alternative, and also the cost associated with all of these phases of developments, plus also economic issues like trials that go into other geographies where they're cheaper and where they're easier to produce, which also will generate potentially an evidentiary gap in terms of the regions and the patients under which those tests are undertaken. Just to give you an idea, for every 100 medical products, complex medical devices that go to market, about 30 of them will ever become commercial, and only 5 of them, of the drugs, will ever become commercial. And that means that from every 100 patients that are participating in those trials or those studies, about 47% of those involved in medical devices will remain with medical devices that are never going to be commercial in their life, in their lives. And from those that have where drugs have been tested, about 68% of those would have been using drugs that will never become commercial. So we are actually exposing people at risk already, even as part of our testing process. I'm not going to delve into a lot of detail, but you know very well that both in devices and in drugs, costs are enormous to get products to markets, and we are going through life cycles of these products from conception to launch of about 13 years on average, which means that at the speed at which innovations are coming into the market, this is very, very slow. Let alone the lengths of trials or studies that require much more long-term analysis. So what if we could somehow have a mechanism that we could build models that provide insights and helps us to anticipate post-market failures early on. And what about if we could optimize through those techniques the designs and therefore prevent those failures before they happen? And what about if we would think about human trials, which, as you will see later, we still believe are important, not be discovery trials in a sense, which is what we're doing now, but be becoming confirmatory trials of our predictions that we have done in modeling and simulation, pretty much like we do in the aerospace sector, where we use a lot of modeling and simulation to come up with new designs of airplanes, and then we build them and then we test once we are heavily convinced that this is, highly likely to be successful. So basically, if you look at this slide, the horizontal axis is the whole life cycle of a product from discovery all the way to post-market monitoring, and the vertical axis is what I call safety signals or inefficacy signals. So prompts that we have that things are not going well, either because of risks or because of things, it's going to be inefficacious as a product. So what we are doing at the moment is very much related to what we do in this, what is shown in this line here, which is that we are doing relatively little testing early on. And by the time we have investment in that product, because most of this part will happen in academic environments, in clinical environments, by the time we have evidence, that evidence will, but by the time, we come to develop preclinical and clinical studies, things that can go wrong will start going wrong. And that will either limit our regulatory approval or will eventually lead to other complications down the line once the product is in the market. Now, the deeper we go in these pipelines, the more expensive is that failure. And my question is, wouldn't it be great if we had some way which we could test in early stages of these products? We could test them in a way that is inexpensive, and therefore we can do it more thoroughly and in more comprehensive manner, but at the same time that we could test those devices with as close as possible to the circumstances that they will have in later stage testing, so that we actually expose those products to the type of variabilities in terms of anatomies, physiologies, patient conditions, patient lifestyles, potentially even use administration by a specialist if it's a product that needs to be delivered like an implant, the variability of expert of the interventionist that is doing the therapy in terms of the learning curve, and we could somehow anticipate what is likely to happen when we go down to the market. Well, that's what this whole idea is about, is supporting both patient benefits in terms of reduction of risk and maximizing output, but it's also about reducing failure in terms of commercial failure with all the consequences it has financially. Now, modeling and simulation, we believe, is also an integrator. It's something that brings together all the different sources of evidence we have, or all the sources of data we have, whether it is real-world data, is bench test data, is animal information, it's procedure information, is information about the device or the product itself, or is about variability of the human that is involved. And that is something that we can extract from various sources of information that we have at our disposal. Now, when we talk about modeling and simulation, we are referring to both the use of AI, not as a device or as a product, but AI as a data-driven approach to do predictions and modeling, but also, and more importantly, to mechanistic models where we actually simulate quantities based on first principles, whether it's physics, physiology, biochemistry, or others. Now, these sort of technologies, and sorry if I'm going to focus a bit more today on devices than on pharmaceuticals, but there is other people in the community that are working specifically on pharma, and there is similar progress in that area, that the one I'm going to show on devices. The use of modeling and simulation in devices has been used historically for forensic analysis of product failures. And later on, the use was seen as valuable, and it was moved to the more R&D cycles of the product. So when, during the design phase, to understand different potential designs. But what it has not happened, and I would say the same is in the pharmaceutical domain, where we use, for instance, modeling to do ADME and dosage analysis, and we also use modeling and simulation and AI in particular now for drug discovery. What hasn't happened in both areas is the integration of the whole life cycle and the early prediction of later stage outcomes as much as I think we are promoting or we're suggesting that we should be doing. So what is an in silico trial or an in silico testing, if we're talking about preclinical? Is a detailed prediction model, whether it is mechanistic or data-driven, that allows us to use highly controlled virtual conditions on virtual cohorts that we build and that represent realistic operational conditions or target populations that we are of particular interest. That could be, for instance, underrepresented communities. It could be particularly vulnerable communities. And what we do is we develop models of the various components and then models of their interactions in ways that they are, as I said, not only data-driven, but mechanisms-driven where possible. We talk sometimes about in silico evidence to refer to the evidence that is generated from in silico models. And if you're wondering why we call them in silico, is because we are considering essentially computational models that run on computers, which are mostly done of silicium, and therefore the word in silico in similar ways that we talk about in vitro, in vivo, in situ. Now, how do we conceive, we as community that are working in this area, conceive the use of modeling and simulation in the future? Are we saying that we are going to sort of, we're advocating about not using bench, animal, and clinical studies? No. What we think is that the question is not so much as if we could replace with computational modeling the other approaches, but as to whether we can sensibly take decisions about medical products on the basis of three sources of evidence: animal, bench, and clinical, when in reality we have a fourth way to do things, which is CMS, or computational modeling and simulation. And what we also believe is that we need to use the right tool for the right purpose in a way that is proportionate to the stage where we are in the process of innovation, and also in a way that is risk-informed evidence, so that depending on whether we are only going to be using modeling and simulation to take a certain decision, or whether we're going to combine modeling and simulation with other sources of evidence, we need to take into account that into the risk profile. We'll talk a bit about more later on about what we mean by risk-informed frameworks. So let me give you just some examples to show that this is not something that may happen. This is something that is just happening. It's just that regulators are not necessarily assessing this evidence today. And possibly trialists are not used to see this evidence that medical product manufacturers have. But this is, for instance, a study. This is more on preclinical testing, where Cyber Biomed, for instance, has developed a simulation tool that tries to replace the bench test of putting their device in a cadaveric specimens and then put that into a machine, into a physical simulator that generates, a year worth of gait cycles to look at wear and fracture of the bones, and this year being an accelerated test that tries to mimic what may happen in the lifespan of about 10 years of the product. So these are very expensive tests, lengthy. We need to wait at least a year to get a sense of 10 years worth of wear. And we need to do that in multiple times to do statistics, while we could actually generate a library of bones with very much more physiological conditions than a cadaveric bone, and expose and calculate the fracture risk profiles or the failure modes of these devices. This is another example. This was a device developed, in silico trial developed in the University of Bath in the UK, where in collaboration with J&J, where they develop a sort of a fixation device for high tibial osteotomy, which is this wedging operation to remove a part of the bone to strengthening legs, and then put the device to keep the system fixed until there is sufficient bone regrowth. And what they did is they did a study where they show through in silico methods that this was likely to be successful, and also an idea about the magnitudes of the effects they were trying to measure. And with that, they then went on and designed a more de-risk human trial that is ongoing, as far as I know at the moment. Also to show that regulators are engaging in this whole, a narrative. This is work by FDA, and is known as the Victory trial, and it's a trial where they are trying to compare two diagnostic modalities, so digital breast mammography with digital breast tomosynthesis, and where here, instead of being a therapeutic device, is a diagnostic device where we replace real humans with a library of anatomical models of different characteristics. We model the physics of image formation associated with that particular device, and then we also mimic the radiologist that is taking measurements by essentially an AI tool that replicates the performance of the radiologist and in front of a given image that is the one that has been simulated from the system. And by doing that, they show, and by developing phantoms, digital phantoms, where they have breasts of different densities, different levels of tumors, and different constellations of those, they show that they could do a comparison that was very consistent with comparisons that have been done in the real world, in other studies, in other real trials. Now, by now, you may be asking yourself, are in silico trials any good in replicating what happens in a real conventional trial? And this is basically the next part of my presentation, where I'm going to show a work that we did in my lab. So one of the areas where I'm working is in hemorrhagic stroke, particularly in the area of aneurysms, and we're looking at different ways of treating this, this disease. You might be familiar that when you have aneurysms, they tend to appear mostly in the circle of Willis around the bifurcations, and you either can do clipping, which requires craniostomy, or you might want to use minimally invasive techniques by going through the groin under image guidance and place either coils with a scaffolding, stents, or flow diverters that actually redirect the flow, and they don't need to use coils. Now, the problem is that there is a ton of these devices for this particular need for this particular condition. And when I'm saying a ton, it's probably about 40, 50 devices that are there in the market. And think each of them, the number of trials and studies that are running independently. And sometimes one wonders, does it make any difference, all of these designs? Are they actually fundamentally addressing the problem, or they are just small variations, in areas where they won't make any difference to the improvement of the disease? And especially, the table on the right here, which is intentionally squeezed so that you can see the amount of them, but not necessarily to read the details. These are all of the products that are out in the market, or there were some months ago in this domain. When you also look at the lifespan of these devices in the market, it's curious, because remember, we said that it's about 13 years on average to come from the concept to the implementation. But the device that we're going to be talking in a moment is what is called the pipeline endovascular device. This was approved by FDA in 2011, so about here, and is responsible for this orangey part. So the vertical axis is the percentage of the market that they are taking over time. And you see that this has started being something very small, but very quickly displacing pretty much all of the other products and pushing them aside. So it's not only that these companies need to develop devices, but they need to keep them in the market for a decent amount of time in order for them to basically recover their investment. And at the speed at which this is happening, you cannot spend 13 years in the testing process, because that probably is even larger than your lifespan in the market. So we're going to talk about this device, which is the flow diversion, is a flow diverter called pipeline endovascular device, or PED, that is currently produced by Medtronic. And this device has a simple mechanism of action. You put a stent, the stent redirects the flow into the mainstream, it stagnates the blood flow, which produces thrombosis, and then that actually removes effectively the aneurysm out of the circulation, but it's not grafted, this stent, so it allows flow to go through the small vessels that are responsible for, feeding the brain and avoiding that you have an ischemic stroke. So this paper here is the pivotal trial that led to the approval of that device. So it took about 8 years from design to publication, 6 years of recruitment, probably 30 to 40 million pounds, and a whopping number of 108 patients. Now, Medtronic claims, and I need to say we did this work unrelated to Medtronic, so we didn't get any funding, or we actually didn't even collaborate with them directly to do that. We did it fully independently. But they claim in their marketing that this is the most studied flow diverted in the world. Now, this trial that I was talking about is called the PUFS trial, from pipeline for uncoilable or failed aneurysms. And the PUFS trial is this one. This is the first one that was done. Then they did Premier trial, which was for small aneurysms. PUFS is mainly for large aneurysms. And then they did Aspire, which is a registry that essentially confirm is a registry, okay? So if you put these all together, there is about 500 patients. That's all the evidence they had, and probably they went to market with this first two, or maybe even just PUF. Now, what we did is we ran a comparison between those three trials, or those three studies, and our in silico trial that we call FVPAS. And I won't have time to go in a lot of detail, but there is a publication that you can consult, and I'm very happy to have follow-up discussions with you. And we also involved Kit Roos, who is a trialist that was involved in our analysis and our assumptions that we did there. But fundamentally, what we did in this study is we created a library based on real-world imaging from patients that had similar characteristics to those of our trials. We virtually implanted the stent in each of them. So we didn't use real-world data of implanted patients, but real-world data of imaged patients. And then we did the virtual implantation through mechanistic models. Then we have a model as well of the physiology, where we can model hypertension and normotension, for instance, conditions, so that we could simulate what happens if all the patients are normotensive, or what happens if they all are hypertensive. And then we solve the flow, the equations of the physics of flow in those physiological conditions for those realistic anatomical models. And when you produce these simulations and then you extract quantitative simulated markers from those, what effectively you have is data as if you would have done a normal trial that you can then analyze and provide analysis in very similar way you would do with any other study. So these are the results. What I'm showing you here is that the FDPAS trial, the overall efficacy was 75%. It took us about 3 months to do this study. These studies, if you put them all together, they run from 2008 to 2016. You see that acute response, so immediately after treatment and maybe a year, they're all approximately around 73 to 86%, about 75%, which is similar to our study. But what we could also do that you cannot do in any of those trials, because they're so small that you cannot do group analysis on hypertensive and normotensive, and nobody will allow you to sort of do a study where you stress test people with an aneurysm. What we could do is simulate conditions which are essentially create counterfactual conditions where all of the patients in our trial are either normotensive or hypertensive, showing that there is already a 15-point difference in that situation, everything else being completely identical. So roughly, to get a sense about what it is, we take an imaging library. So you go to your clinical colleagues or your NHS records, you extract patients with imaging of brain aneurysms in a way that is, consecutive. Then you impose into that large data set all of your inclusion and exclusion criteria very much the way you the same inclusion and exclusion criteria you use in your trial. And then there are techniques, and there are plenty of publications about how to do that, about how to virtually implant those devices into these anatomies. So you have real world anatomies, real physics-driven, mechanistic-driven deployments. And then we have to develop some biomarker or some endpoint. We talk about technical endpoints, which is effectively a surrogate endpoint. So these are things that we can simulate and measure. In these cases, the reduction in blood flow within the aneurysm, which is the reason why the clot is formed. Because we cannot simulate, complications or death as a marker, but we can get signals of complications or signals that are likely to link to that. You could also create physiological models of the boundary conditions that we call, or in this case, the blood flow regime, that represents, for instance, the blood flow in the carotid artery in a group of 100 patients, in this case, or 100 subjects, I should say, in both rest conditions and stress conditions. And how we do know that? Because they were measured through Holter systems for 48 hours, and we develop a model from where we can know what are the average behavior in more relaxed periods of their day and in more stressful periods of their day. And then finally, you can actually put every single device to every patient, so you don't need to do randomization for that purpose. And you can compare, say, two commercial devices, Enterprise and Pipeline, and how each of them affects these surrogate endpoints that we talked before. Now, the vision is to do that not with a few patients, but to be able to use data sets from our growing regional care records so that we can actually build populations that after applying inclusion and exclusion criteria are, say, 2 or 3 orders of magnitude bigger than any of the trials of the data that has fed the trials that we've seen. So in that sense, they are more thorough and more exhaustive than a regular trial. So for instance, we are working with list teaching hospitals and doing a data extract for the last 10 years of all of the patients that they underwent brain angiographic imaging. Again, many of them will not have even aneurysms, and many of them will not have been treated of aneurysms, even if they had. But what we are trying to do is a population characterization of the variability of, say, anatomy and other demographics. Now, when you do that, you start also seeing a number of issues, like some of the inconsistency we have in our healthcare records. Like, for instance, nobody would believe that these patients have only 2% diabetes, because this is self-reported diabetes, not the real one. So we need to be careful about the quality of the data we use. But you also see some of the problems that even in our NHS, there are limitations in terms of how represented every ethnicity is in there, which will also have implications on the representative of our trials. We also, and I'm not going to present this today, we're working with industry in other areas like TAVI to actually compare simulations with head-to-head comparisons with real randomized controlled trial data. And we're also working with data from UK Biobank to build populations of organ anatomies that are not limited or don't have the selection bias properties of, all my subjects have gone to the NHS, for instance, which means that there is at least some reason for them to think that they might be ill. And then more recently, with developments in the UK about developing the Health Data Research Service, the idea is that we might be able to do this at really national scale. And of course, as you do that, some of the issues that I'm sure that you might be worrying yourself about, biases on the data that feeds the models, will be addressed. Another example is the use of in silico trials for off-label use. So this device that we discussed before is only approved by FDA in a particular region, but we want to look at what happens in the posterior communicating artery, which is outside of the approved regions by the FDA, and where unfortunately we have 25% of the aneurysms on the brain. So, there is a case where patients come to interventional radiology saying, I have a problem, and then that person needs to be treated. Maybe there is no product in the market. So we did work in the past where we show that actually using these sort of in silico techniques, we can identify the performance of these devices in the off-label use. Now, one small diversion that I need to explain to you is that if this is the circle of Willis here, and this is the posterior communicating artery, which is the one where we want to look at the uses of these devices, the off-label use, you need to be careful that the posterior cerebral artery, which is, sorry, this one here, it comes in different flavors. So you could have the artery, you could have, which is called the non-fetal variant, or there are two fetal variants, one of which is that you miss completely this segment, and this is normal variation, or you have a hypoplastic segment, which is thinner than the natural one. So if you're going to look at what happens in the aneurysms in this area, you need to look at what is the impact that these other characteristics will have imposed. So because we understand these and there is sufficient data in the literature to look at what are the changes in diameters, the changes in flows, we can actually generate those chimeric or counterfactual scenarios, and here is the data. So if you look at the non-fetal variant, and I think I mix up the case, this is the non-fetal one, and this is the fetal one, the fetal variations. The non-fetal variant has an outcome of 88% in terms of occlusion rate, which is our endpoint. However, the fetal variants have half that outcome, percentage of efficacy. Now, this, you may say, well, let's not use it in fetal variants, but actually those fetal variants there is some prevalence of those. And most importantly, they tend to be more present in Black and in Hispanic populations than in white Caucasians. So this also is telling you that this device is essentially not being equitable in the care that it provides to the different ethnicities. One final example is the use of these techniques for stratification. So I'm going to change a bit gears from another device. These are coils, which is another device for the same type of problem. And one parameter that you use is what is called the packing rate, so how much of these coils you push into the aneurysm. Of course, the objective is the same, slow down the flow, producing clotting. Now, this packing rate, which is usually measured as the percentage of volume of the coils over the volume of the aneurysm itself, we have developed, we've done some publications where we show how this packing rate changes the hemodynamics of the aneurysms. And I'm going to show you some of the key ideas here. So what you're observing in the left picture is a plot that shows models that we developed for the coil. So I'm not discussing how we model the coils, but if you trust me, we put those coils into the aneurysms. And each of the columns here represents a different packing rate of the aneurysm. So 20%, 25%, 32%. Every color is a different coil. And we put increasingly more coils. Now, this configuration A, B, and C that you see here is equivalent to, say that we take the 25%, the middle column. Configuration A is we insert coils with our algorithm, we then take them out, we put them again, then that's configuration B. We take them out, we put them again, that's configuration C. Because these coils are not always exactly falling in the same position, mimicking the behavior of the interventionalist, what you end up having is multiple alternative treatments of the same subject. And what we have observed is that if you show the amount of flow reduction through the neck as a function of the packing density, the higher the packing density, the higher the flow reduction. But there is a point upon which you can keep putting coils and you're not going to change the flow anymore. And in fact, if you keep putting coil, what you're doing is wasting money because they are expensive. B, you're potentially pressurizing those aneurysms and bursting them, which is dangerous for the patient. If you put aneurysm coils below this threshold, the problem is that then it depends a lot more the exact way in which you put that, meaning the particular interventionalist within the operation. So there seems to be, for these devices, a sweet spot where they are optimally effective, minimally expensive, and minimally reliant on the expertise of the clinician. And imagine now that we run this across a large population, so we could figure out that that sweet spot is what we need to personalize. And most importantly, that we might not need a different type of coil, we might need to know how to use it better. So a study that was done about 10 years before our study showed that essentially what we are showing here in a lot more detail, then stable aneurysms tend to have a higher packing rate than unstable aneurysms, and that this packing rate that is optimal depends on roughly the type of aneurysm, whether it's large or small, and then the neck size. Okay, what I show you is just a tip of the iceberg. There are tons of other uses of potentially in silico trials that I'm not going to get into today, but hopefully I will have raised your interest in this to explore and look for them. Now, from a regulatory point of view, are we ready for prime time? So I would say this is hotting up, and I don't think we can ignore it anymore. So there is a lot of documentations by FDA that is pointing into this direction, and by EMA. Next week, there is a modeling, this week, is a modeling event in Brussels on drugs, run by EMA. In a few weeks from now, there is a meeting in Washington on medical devices run by FDA on modeling and simulation. There is documents and strategies where they are pointing to the use of modeling, and by modeling I mean this type of modeling, increasingly higher. And you can see in the slides when we share them with you some of these documents. Even in the clinical guidelines from some of the regulators like TGA in Australia, there is already reference to the use of modeling and simulation in certain case studies. Okay. Now, we do need to come up with principles for credibility of those models. And a group of us is working on developing a framework for those, of what we call more generally model-informed evidence. And just to give you a gist about the sort of inputs that we are using to think about this, this is what is known as the Aqua book. It was developed in 2015, about 10 years ago, by the UK government in an unrelated to health domain, but where they talk about analytics models for decision making in government, so more general case than the one we are looking at. But it's interesting because they have come up with a number of principles like proportionality of evidentiary response to to the phase where we are, assurance throughout the life cycle is not just in one point, the importance of validation and verification, and also what they call analysis with rigor, that stands for repeatable, independent, grounded in reality, objective, that we have understood and managed uncertainties, and we have addressed the questions of interest robustly. There is also frameworks emerging by BSI standards, and Joy State is one of the key colleagues and members in our consortium that is developing those for what we call proportionate and adaptive governance of innovative technologies, and how we may use in silico trials actually to rethink the sort of governance, the regulatory governance in this domain. And also an increasing number of documents and regulations that are starting to appear across all of the geographies. That has also raised the interest by forums like HCI and AMDRF. So I want to conclude by saying that in order to make progress in this area, we really need to develop, we need to co-create essentially new ways of working between regulatory affairs people, industry, academia, and that's why your community is so important, as there are other communities as well to do this. But one of the things we've done a couple of years ago is develop a survey where we map what are the barriers and enablers for in silico technologies. And the top three things that came up is the need of more confidence in regulators to accept this technology, so there is less uncertainty on industry that this evidence is going to be accepted. Second, the importance of developing credibility frameworks, which we've been working hard since. And also the next two, which relate to this webinar as well, is the importance of developing skills and awareness among regulatory affairs and also among industry, because it has been identified as an area where there is shortage of skills. So we put together a consortium, which is what we mean by CERSI. And I'm glad to say that we do have colleagues from Ireland as well, and I think I've seen Ted Vaughan on the call before, who is our proxy in Galway. and we would very much like to expand that relationship. We believe that there is a lot of opportunities given the strong industry base that you have in Ireland in this area to work together. So what we've done is we focus in this analog for the first year, which is we're halfway through it, in what we call in an exploration about the current international model credibility frameworks and developing a problem-based approach to learning as a community. I don't have time to go into detail in this, but what I want to say is that there are 10 case studies that we're running, all of which are led by industry, and all of them are based on real questions of interest and context of use that are industry-based. Each of these groups has two companies, sometimes competitors between them, two regulators, two academic institutions, and one member of the lay member that is participating and feedbacking. And we are developing some documentation that will be produced over the coming months. So what we essentially are doing is we are taking standards like ASME BMB40 standard and applying this process through those devices and looking at what is the good, the bad, and the ugly of applying them, and what are the things that we need to learn. But fundamentally, this framework says that there is two aspects. There is a risk-informed framework, and it says that there are two considerations for the risk. One is the model influence, so how much your ultimate question of interest will be answered purely on the basis of the model, or is it the model plus a lot of other things? Of course, the more is only on the model, the more risk there is. And also what we call decision consequence, which is, if you get it wrong, what is the worst thing that can happen? It's just inconvenient the patients, or it's actually death, right? And I would argue that you need to have this risk-informed framework for any other source of evidence as well. So what we are doing with that standard and with the subsequent guidelines that were developed by FDA is to explore their use, explore also some of the document that has been produced in the literature about good simulation practices. These are two books that you might want to explore, one already published, one about to come out. And we're drafting together with MHRA a white paper, a position paper, that it would be great to have some feedback in due time from colleagues from Ireland as well, because we hope it will be fundamental principles that we should be able to agree upon. And they're going to be focused primarily on principles about the quality and the credibility of the model themselves, of the data that fits the model, and on the process of generating inferences from that model as well. So with that, what I like to say is that this is in motion. We think that it's going to be a transformational new input into the community. We acknowledge that there is a need to both transform, it's both a technical but also a cultural transformation. It's a different way of working in many ways. And that is quite important to bring all of the stakeholders, including patients. Simon suggested that I may want to tease you with some questions. You don't need to answer them now, or we don't need to cover all of them, but just the questions that I would like to put to your community. I'm sure you have questions for me as well. Is how do you envision we could integrate these sort of techniques into clinical trials protocols? And what are the challenges that you see in aligning model outputs with clinical endpoints, what I call technical endpoints and clinical endpoints? Is there data that you are collecting in trials that are a bit underused, and that we could maximize the use of that collection by, through modeling and simulation, generate new forms of computational evidence that goes in hand with yours? How do you as we are asking some questions to regulators, that what do you consider the hurdles to be regulatory in terms of the acceptance of these approaches, and what methodological innovations we may need to do to affect them? And then finally, and more broadly in general methodology of trials, but maybe thinking here in adaptive trials, is there a way that we could think about using this evidence as a way to adaptive trial design, or that when there are early signs of risk or complications, rather than waiting for further events to accrue, that we could almost start studying through modeling and simulation what if scenarios about what would happen if that was actually a much more not a one-off, but a more sort of consistent signal. And with that, I'd like to thank you for your attention, and like to take any questions. You will see at the end, this is a slide where you will have a number of resources that we're putting together where you might be able to explore more in this area.